Welcome, everybody. My name is Angela Collins, and you are now in a webinar entitled Exploring the Potential of Macroalgae, otherwise known as seaweed, aquaculture in Florida. So if you are not in the right place, now is your chance to leave, but otherwise we'll go ahead and kick this Zoom off. Thank you all for joining us today. Today's webinar is focused on a recently funded project that is exploring seaweed as a potential aquaculture product in Florida. Throughout our program today, you'll be receiving a wide variety and a broad spectrum of information. You'll see a lot of links within the presentations, but just please sit back and relax because we're recording today's webinar, and we'll also be providing a list of the presentations as well as resources and relevant links in a PDF file to everyone who's registered. So you can feel like you can just relax and you don't have to remember all of the things we say because we'll provide you with extra resources after the meeting is over. Due to the large number of registrants that we have, we just ask that everybody stays muted. If you have a question, feel free to type it into the chat and we'll answer it as we can. You will have a chance to ask questions directly to the speakers after each one of the presentations. And in that case, you can either raise your hand so we know to unmute you, or you can put your name in the chat and we can go ahead and call on you. So there are lots of ways to interact with folks as you have questions. Just remember that we are recording today's presentation, so your voice, if you ask a question, will be recorded. Okay, so this project is funded through the NOAA Sea Grant Aquaculture funding in fiscal year 24. And we have a pretty fantastic team. This is the team, and you're going to be able to talk to all of these folks today. Our fearless leader is Dr. Ashley Smith. She's a biogeochemist in the School of Soil, Water, and Ecosystem Sciences, and she's located down in Homestead at the Tropical Research and Education Center down there. Gabby Forza is with us today, and she is our graduate student who is leading the charge on a lot of the field work as well as a lot of the laboratory analyses. She's probably the biggest lover of seaweed that any of us have ever met, and you'll get to hear from her as well. Dr. Aaron Welch is our industry partner. He's been working on exploring the potential of seaweed aquaculture in Florida for quite a few years now. He's the owner and operator with his family of Two Dock Shellfish, and he's located in the Tampa Bay area. Dr. Dale Laughinghouse is a phycologist. He's located at the Fort Lauderdale REC. He's probably one of the biggest microalgae lovers we know, but he also likes the larger version. And so he's going to have some really interesting things to share with us today as well. Dr. Andrew Ropicki, he loves money. He's our economist, and he's a marine resource and economics professor in the Food and Resource Economics Department at UF, and you'll be hearing from him. And again, my name is Angela Collins. I'm an extension scientist, and I am based at the Tropical Aquaculture Lab in Ruskin, which is in the Tampa Bay area as well. So just to kind of give everybody a snapshot real quick on what we're talking about here, seaweed aquaculture on a global scale has tripled in the last 2 decades, right? It's one of the fastest growing commodity sectors worldwide in the aquaculture industry. Asia currently dominates 99% of this market. Most of the imports and exports are taking place in the Asian market, but it is a crop both for things like food. A lot of times people think immediately of food, but it's also used in pharmaceuticals and nutraceuticals, as well as thickeners and biopackaging, fertilizer, animal feeds. There's lots of different applications for seaweed as a commercial product on the global scale. It's also been quite recognized for the ecosystem services that different seaweed may provide. And so it's been explored in several regions and celebrated for its environmental benefits and water quality and restoration aquaculture initiatives. I should mention that there are over 12,000 species of seaweed, probably more. That's probably a very big underestimate, and only maybe 27 species are actually cultivated at a commercial scale worldwide. In the United States, it's very much still an emerging industry. It's very young. Most of the production that is occurring in the United States is in the colder water, right? Think Pacific Northwest. Think the North Atlantic. These are like kelps, the larger colder water species. Most of these operations are relatively small in scale. Most of them are seasonal, and they're almost all reliant on pre-existing working waterfront and fisheries infrastructure that's already in place, which is why it's so exciting to think about Florida as a potential place for opportunity of growth for this sector, because we do have such an expansive coastline. This map shows where seaweed aquaculture is currently taking place at some scale within the US. You can see not very many states are highlighted. Most of them, again, are in the colder water temperatures up there in the Pacific Northwest and New England and Alaska. Florida is highlighted, but the culture, it's worth noting, that's happening in Florida right now is in tank systems, right? It's enclosed, it's at a very small scale, and in tank systems. So really in Florida right now, there is not a ton of seaweed farming going on. Right now, there's still a lot of information that's needed on what species are even viable for commercial utilization. Are there regulatory hurdles? Is there a market? Is there processing infrastructure that's actually feasible and is going to be realistic to implement in our great state? But we do have this beautiful, extensive coastline. We have existing working waterfronts and a thriving shellfish aquaculture industry. So there is this potential for industry collaboration or potentially alternative markets if there were seaweeds that could be cultured economically and realistically feasible. There have been multiple projects over the last 5 years that have sort of explored some of this potential of seaweed aquaculture in Florida. And those projects are listed on the left side of your screen there. Dr. Aaron Welch has been involved in all of these from the beginning. Him and Loretta Roberson up at Woods Hole looking at tropical seaweeds back in 2021, and they did some small scale pilot studies in the Tampa Bay area looking at seaweed culture. Nicholas DiLorenzo, who's at the University of Florida, along with Ashley and others, have been exploring the potential of using compounds that are found in seaweed as an additive to livestock feed, because that it has been noted that some seaweed compounds can be added to livestock feed and actually reduce that animal's carbon or methane emissions. And so there have been projects that UF has been involved with over the last 5 years or so that have started to, kind of tease out some of these bits of information that are required before you can really develop a market at any scale. Today, we're going to be talking about the most recently funded project. And again, that project is entitled The Current and Potential Role of Macroalgae Aquaculture in Florida. And that's going to be the focus of today's talk. And you're going to hear from all of those researchers, as I mentioned before. So today, again, let's just practice as active listening as we can. Stay muted. If you have a question, as a repeat for those of you who just joined, if you have a question, please type it into the chat, and there will be an opportunity to unmute yourself and ask questions after each speaker finishes. And then I just wanted to remind everyone that this is definitely an opportunistic managing of expectations with seaweed aquaculture, right? This is just one of the first steps, and research is, going to inform any potential future production. And again, as a follow-up, today's webinar is recorded, and you will be provided a link to the recording as well as a list of resources if you want to really follow up or get more information on some of the projects that are happening currently within the state. And with that, I'm going to go ahead and stop sharing my screen and pass the microphone to Ashley.
Awesome. Thanks, Angela. Let me just get my slides situated right here. Let's see. All right. Are you seeing what are you guys seeing?
We're seeing your title slide.
Great. Perfect. Awesome. Well, thank you all so much for coming today. I'm Ashley Smith, as Angela said. I'm an associate professor and a coastal biogeochemist. So as a biogeochemist, I think a lot about nutrients and how nutrients move between sediments, water, and air. and then the role that human activities have in these exchanges. And this is really what led me to our current project and also my interest in shellfish. Seaweed, shellfish, they are all part of our working waterfront or could be part of our working waterfronts. And the addition or subtraction of these from the coastal ecosystems has consequences for the way nutrients are cycled. And that's the lens that I'm thinking about when I think about seaweed. So it's really looking at seaweed as this extractive service that can help support our working waterfronts. So with that, I will say I am not an expert in seaweed, and I'm so thankful that we have a very large team who are experts and think a lot more about seaweed. Until we started this project, we mostly just had shellfish in our lab. So I just love expanding on our work and looking more at how we can help, what we can do in Florida. So our project right now is really focused, and we'll explore more today, why seaweed matters and what success looks like from this project, even if we find out that seaweed is not a viable product for Florida right now. All right. So Angela already covered this a little bit, but I just wanted to take a little bit of a deeper dive into our project and why seaweed and why now. When we started writing this proposal, it came to us as this opportunity to diversify Florida's shellfish industry and what products they were having. We all know there's a big clam aquaculture industry, and we're really good at growing clams here in Florida, but there's also the chance to expand beyond that and to kind of make use of what we know, and seaweed presents one of those opportunities. And seaweed has a lot of interest on this global market. There's a ton of opportunities for seaweed, food, fertilizer, aquarium trade, other markets like carbon offsets, biofuel, cattle feed additives. There is a lot of interest in growing seaweed and things that need seaweed. One of the benefits of seaweed, and again, where I'm thinking about this, is that seaweed has this chance to provide environmental benefits or ecosystem services. Seaweed works like a sponge, and it sucks up everything that's in the water, including nutrients like nitrogen. And when we have too much nitrogen in our coastal ecosystems, that's when we turn that water that guacamole color green. We don't want that. So seaweed can actually take up some of that nitrogen and prevent it from feeding other algal blooms. And then when that seaweed's actually harvested and removed from the ecosystem, that represents a complete extraction of that nitrogen. I'm also really interested in seaweed in Florida because we have the opportunity to culture products year round. Although sometimes it might get a little too hot, but we don't have to deal with that cold snap or those freezing conditions that they do in those colder places like Angela mentioned. And so this does present a lot more opportunity and something that really needs to be explored. So why seaweed, why now? We have this opportunity to diversify our shellfish aquaculture industry. We need water quality tools in Florida. We have this chance to really do a deeper dive and fill in this gap in our knowledge on seaweed production. And there is this global interest right now. When I started thinking about seaweed, I was actually a postdoc at the Virginia Institute of Marine Science. And many of you on this call might have heard this story, but through the clam lens. But one thing that we observed when we would go out and sample our clam sites was that seaweed was just accumulating on these nets and on the shellfish gear. And that our partners would have to go out and actually physically remove all of this biomass. This is a photo from Virginia, so it might be a little different from what we do in Florida. But I remember being out on the water and seeing our partners use like an underwater lawnmower to move off all of that macroalgae. And then they detach it from the nets, and then it just sort of floats off. And I remember thinking to myself, like, that's a ton of nitrogen that they're losing. So we actually did a little study, and this is published by Annie Murphy in 2015. And you can see here in this figure that the red bars are our clam sites, our clam aquaculture leases. And then we have clam leases where we have that macroalgae, and that is that green bar. And this is the ammonium production. So we get ammonium production from all of life. And what's happening, and what I love about this figure, is it shows this decrease in the ammonium regeneration into the water column with the presence of that macroalgae. So to me, this means that the clams are there, they're doing their thing, and the macroalgae are actually acting as an additional nitrogen sink, and they are now storing and sequestering all of that extra, all of the nitrogen, not all of it, but a good portion of it. And they're actually being this really good nitrogen store. So they really prevent nitrogen from returning to the ecosystem. So having these data and seeing what was happening in Virginia really made me think about the importance or the opportunity to integrate macroalgae and seaweed harvest with clam aquaculture as a way to be effective at reducing eutrophication and as a tool for water quality restoration. This is one angle that we're looking at within this project, but for our project in particular, what we're really emphasizing is trying to find out if seaweed aquaculture is a good fit for Florida. And I want to emphasize that the answer to this, any answer, is actually really valuable. So the core question, is seaweed a good fit for Florida? But we're not just looking for that yes. What we're looking for is really clarity and understanding of how best to move forward. But let's say that our answer is yes. Let's say we do all this work that you're going to hear a lot about from our next speakers, and we figure out that seaweed has a lot of opportunity here in Florida. If that's the case, what our project will be able to help do is develop cultivation protocols. We'll have assessed the market potential. We've identified ecosystem services provision, and we'll be able to help support growers through economic diversification of their product. So if the answer is yes, seaweed is viable, this really just opens the door for new products, water quality improvement, and economic growth. But the reality is that the answer could also be, it's not right for right now. So what if we find that seaweed aquaculture here in Florida is not the best opportunity at this moment? There's a variety of reasons that this could be. It could be species limitations. It could be market barriers. Maybe we don't have a species that is actually has a high return on investment. There could be environmental issues. That's all fine. The fact that we might find out that it's not the right opportunity right now is still a win. It means that we've saved time, we've saved money, and we've saved effort for growers and policymakers by helping them identify where they should be spending their energies in the future. But what we will end up coming out with from this project regardless is valuable data that's useful for restoration going forward or planning going forward. We're really producing new knowledge that will help Florida's aquaculture industry make the most informed decisions as we go forward with whatever solution, whatever the answer is to this flow chart here. So now our approach to addressing our central question, we've designed this in a multi-structured, multi-prong approach. The first is that we want to identify dominant seaweed species that are naturally occurring at shellfish leases. We're really focused on what does already grow naturally and recruit naturally in Florida. This also helps us focus on candidate species that are already adapted to the conditions that we see here in Florida. Next, we're going to measure their nitrogen assimilation rates, and this is how much nitrogen they absorb, and this is really the key for understanding the potential as a water quality tool. We're also going to optimize cultivation practices in the lab to hopefully improve efficiency on the back end when we take this to a larger scale. And we're really figuring out what best conditions to support best growth for best products or best nitrogen uptake. We'll also be evaluating the market potential and answering, can these tropical species be used in food, cosmetics, or pharmaceuticals, or some other use that we haven't quite identified yet? And then to tie it all together, we're going to do an analysis to figure out feasibility and scalability. But all of this, throughout this whole process, we will also be engaging with you, everyone who's on this call and everyone who's interested in this project throughout our time. This includes the regulators, public, and anyone else who might find this information useful and actionable. I should note our project did start a little bit ago, last fall, but we had a really slow start. As most of us know, we had a couple hurricanes that delayed our actual start. So we are really now in the weeds, so to speak, and getting our first rounds of data back that we can share. So beyond answering these questions through these objectives, the project is really geared at generating these tangible products that have direct benefits. First, we're going to create a species ID list or database that catalogs the seaweed species that we find at shellfish leases, including their traits and potential uses. We will also have that nitrogen uptake data, so now we can really do full nitrogen budgets at our shellfish leases. I might be the only one who's that excited about that. But it really helps us identify the role that they have in water quality improvement. And we will have that opportunity to help understand how to optimize growing conditions to get the most out of our tropical seaweeds. On the economic side, through this market analysis report that we will actually be producing to give out, will help gauge consumers' interests. And really, I will also come up with tools necessary to help identify, should I invest in seaweed? What are my inputs? What are my outputs? What's my possible return on this investment? And finally, again, we'll produce fact sheets, educational tools, we'll have workshops, and we'll make sure everything is accessible so that this information, it gets out beyond just us. And gives everyone else the chance to really stop and say, hey, I wonder about seaweed. Now they'll have resources to go to when they're thinking about seaweed here in Florida. From all this work and the busy 3 years we have planned, we hope for some core outcomes. And the first is that just having this data to really inform the development or not of seaweed aquaculture here in Florida. We'll also be able to have this really nice link between seaweed aquaculture and clam aquaculture and water quality goals. And then we'll really have tangible tools and products to help identify costs, benefits, and challenges into expanding into seaweed aquaculture. Our goal is to help with this decision and answering this question, is seaweed viable for Florida? Here's what we find, this is what we think, and this is what we know, and put those lessons back into the hands of people who can use it. So what's next? Over the next few years, we'll be diving deeper into each of those research objectives that I've outlined. And we'll really be working, you'll be hearing a lot more from us now as we're trying to understand the feasibility of seaweed aquaculture here. But what's next in the next little bit of time we have together is that you're going to hear some talks from all the colleagues of mine who go more into details about the methods we'll be using and some of our early findings. We're really excited to share with you today what we've learned so far and hear some feedback from you that we can take going forward, because there's still a lot of opportunities to modify our approaches, grow our approaches, and we're curious to learn from you all too. So with that, I thank you all for your attention, and I'll be happy to take any questions.
Thank you so much, Ashley. That was great. And actually, I'm thinking that probably the most logical thing is for Gabby to go ahead and present, and then you guys can entertain questions after Gabby speaks.
Great.
And in the meantime, as you guys think of questions, please feel free to type them into the chat.
Hi. Sorry, I just needed a second to get situated. So hi everybody. My name is Gabby Forsa. I am a PhD student on this project, coincidentally co-advised by Dr. Ashley Smith and Dr. Angela Collins. They did a really good job at summarizing kind of where the state of seaweed aquaculture is right now and where we're at in Florida. And I'm going to be talking about and also Ashley addressed like our objectives, and I'm going to be talking about how we make those objectives happen. How are we getting from point A to point Z at the end of the third year? And with that, we'll get started. So how are we going to identify the feasibility of seaweed aquaculture in Florida? So what exactly do we need to be looking for, and what analyses are we going to be doing to kind of answer some of these questions that we have? And so what we've decided is we need to identify what species are actually in Florida, what native, naturally occurring either through accumulation, or whether they're growing there on the leases are occurring, and figuring out what's there, not just because there's this big jumble and so many things look similar. And so it'll be really good for us to know those. So we can, one, hone in on what are our important seaweed species, but also later for permitting aspects if we get there. And then we need to figure out what these seaweeds are good for. So obviously they're good for a lot of things. Seaweeds provide a lot of ecosystem services, habitat, all this stuff, but in terms of their marketability and their extractive qualities. So in terms of, is there a market for them? So do the seaweeds we have in Florida contain any kind of high value compounds that we can approach a market with and yeah, make some money off of? Or, we need to figure out how much nutrients are these seaweeds actually pulling out of the water? What are their extractive nitrogen capabilities, so that we can quantify that? And then how are we going to connect the industry to this? So obviously the things that we find and address are going to have to be relevant to industry. And then how do we make all of the data that we get actually digestible for people who want to use it? And so I get to talk about the really fun stuff, which is what we actually do in the field and in the lab. And so this is what our fieldwork kind of looks like. So what we do is we go out to our site in Tampa Bay. It's a couple leases, and they currently have, it changes over the course of time, but they have product clams on them right now. And we will go quarterly to these sites. So we get these seasonal variations. And we'll put down a quadrat or 8, and we'll throw that down, and we'll look and we'll see what are the predominant species that are kind of in there. We'll collect everything within the quadrat, and then we will get the excess water out, take a weight, and then that will be processed later in the lab, which I'll talk about. And yeah, and so we will also, so that's the main thing we do at the site, but we will also collect water quality data, which is like pH, temperature, salinity, all these factors that contextualize the seaweed that we're collecting in terms of what is occurring here, what is recruiting at this time of year under these conditions. And also, if we're cultivating in the lab, we want to be able to replicate those conditions and also manipulate them later when we're doing optimization trials. And so here I have, all this work is done through snorkeling or free diving. So there's a picture of us snorkeling in the top left. And then as we go around, you see some seaweed we collected actually earlier this month. And then there's Aaron doing a good job at his job. And then there's me with this rake we had created that was inspired to collect some extra biomass for our analyses. And then there's this little video of what it looks like when we are in the field. So you can have all the plans you possibly want for field work, but sometimes you have really low visibility and you are free diving. So if you got a little dizzy watching that, imagine how I felt. And then, but yeah, so that's kind of what our field work looks like. And then this is what my brain currently looks like. And everything, all the analyses we go through, post these collection events. And I know there's a lot on this slide, so I'll give you kind of a cheat sheet here. These are some of the things we're looking at. So I addressed that we're looking into identification. So if we're doing this mostly morphologically, using whatever resources we can to identify them. I'm also pressing the seaweeds to build out this herbarium. I'm doing some chemical preservation for later, just so maybe someone more well-versed can look at it. Where a lot of these seaweeds, I don't know if you've ever gone to the beach and taken a handful, they all look the same. So microscopy is a really good way to kind of get at the nitty-gritty of those and see the differences. And then the best way is the DNA analysis that Dale is going to be talking more about later, but I do the pre-processing for that. And so the DNA analysis is really important because not only does it confirm the species that we are identifying in our lab, but it helps us kind of narrow down, if this species that we have is similar to another one that already has an identified, high value aspect to it, that we can hone in on that better with the seaweeds that we have. That being said, in conjunction with that, also pre-processing for these compounds, trying to identify something that might have high value that makes it worth it to either be, sloughing this off your lease all the time, or, if someone wants to cultivate this later, which are the ones that they would want to cultivate. I'm also pre-processing for, our nitrogen analyses. So that's basically looking at the nitrogen that is assimilated into the tissue. So how much either the seaweed is taking up, or if we're removing that seaweed from the system, how much are we taking out? And so quantifying either those nutrient credit trading opportunities, or just seeing our environmental impact. It's also relevant because we are doing some growth trials. We've been culturing since, whether Ashley wanted me to or not, since I got here in 2022. And recently we've been doing more focused growth trials and looking at how nutrient additions will change biomass production. And we'll be doing more of those as time goes on. Yeah, and so to get a little bit closer of a look on some of the things we're focusing on. So very passionate about this part. So the ID guides for Florida are not very user-friendly. There has been work done for Florida seaweeds before. A lot of it was in the 80s. I don't know if you've looked at a textbook from the 80s. It is kind of hard to follow. There are some other guides that are available, of course, but they're also specific to their own uses. And so we are trying to create something that is industry specific and specific to Tampa Bay, and because that's where we're starting. And so of course we're going to miss some things, but our main focus is what an industry member in Tampa Bay is going to be looking at right now. And so we're going out to the field, we're doing this morphological review, we're doing everything possible to, create a timeline with the pressing is basically what it is this herbarium, so that we know now what we're seeing, but someone in the future can catch these trends and see what, and someone who is going out in the field would be seeing over time. And then again, ultimately with this DNA confirmation, that is what a sample of seaweed looks like after I'm done with it. It's a lot less cute. Some of the very preliminary data of what we've been running into, especially this last sampling, was a lot of these seaweeds that I've had listed here. Earlier in the season, we had more of 2 and 3, so early in the year, and then this last sampling was more of number 1. And this figure on the left is kind of the distribution we see for Florida, the seaweeds we see in Florida. So this is reds, greens, and browns. And it's great because that is exactly what we saw represented in the seaweeds that we are collecting. All of these seaweeds that are on the right are red seaweeds. And then to go more into our uses. So these uses, again, can be commercial or restorative. So even if we don't find a market for this, we still get to identify, how these seaweeds impact the environment in terms of like their nutrient uptake capabilities. And so we need to identify, do they contain any high value compounds? And then if so, what compounds? And then how can they be used to improve water quality? And so through doing this, we get to learn like an idea of all these seaweed species we've identified, which ones we can hone in on. And I forgot to mention, we are shooting for the top 5 most abundant based off of our data. So I only listed 3, but I'm still working on the ID, that's why. And so we're looking for these top 5 that we can hone in that are abundant, do well, growing, but also have these either, really quality compounds, or have, really good capabilities of restoring water quality. And then we have our industry connection. So I say even if interest exists, which it clearly does, because you're here. And also, this project wouldn't have been funded without interest. Our previous seaweed project wouldn't be funded without interest. And so we have all this interest, but we don't quite know how to do it for our tropical species. We don't have these aspects of optimized cultivation. What infrastructure do we need? What is our environmental impact of either growing or removing seaweed, and all these other aspects? So that's kind of what our project is shooting to figure out, which is, this big knowledge gap that we have and figuring out this feasibility. And so, we want to work with our industry partners to get the feedback of what are their issues, and, is our approach actually helping them? There's aspects that we can do more readily than someone else. So like cultivating lab conditions to optimize, this biomass production and this compound production, and figuring out, what are the best cases scenario for that to transfer that information to someone else, and producing these field guides and these protocols. So we're doing all this work, behind closed doors over here, and then getting to disseminate it outwards to help other people who are interested. And so how will our work inform the expansion of seaweed aquaculture in Florida? So we're going to be, just to recap, we're going to be identifying the species that are present in Florida, in Tampa Bay. We're going to be able to make these recommendations on the high value compounds. So, things that can be extracted from the seaweed that might have some sort of, economically viable reward from them. And we're also going to be quantifying these extractive abilities of the seaweed. So again, their ability to take, nitrogen out of the water column and, improve that water quality, limit those algal blooms that Ashley was talking about. We're going to be, learning how to optimize cultivation. So you're like, okay, I want to grow this. I don't know how to grow this. So, we're shooting to figure that out. And then I think, quite possibly, at least to me, the most important aspect of this is creating these informative resources. So to me, none of this work that we're doing matters if someone else can't use it and learn from it. And so, whether that be someone who's Googling seaweed and the only thing they find is, sargassum washed up on the beach. Florida is so much more than that. Whether it's someone who is going to continue the research after me. I want them to be able to start from where I left off and not start at zero, like I've been doing with a lot of this, not zero, but low on the percentage with all this seaweed. And then also for our industry members to be able to know what they're working with and not waste their time like targeting all these other things, and hone that in for them. So with that, I want to say thank you everyone for your time. Like I've kind of gotten at, we've just started with this project. If anyone has any feedback or any methods they've learned about, they want to share, feel free to email me. But yeah, thank you everyone again.
Thank you, Gabby. That was great. And I just have to admit that I gave a little bit of false advertising by giving people the idea that they could ask questions after each talk, because we're actually, in the interest of time, going to feed right on into Dale's presentation, because he had an emergency thing pop up meeting that he's got to be at 3. So Dale, if you want to unmute and share your screen, we'll dive right into your presentation. And then if you guys have questions for Gabby or Ashley, go ahead and stick them in the chat right now while you're thinking about them, and we promise to come back to them.
Angela, are you seeing and hearing me?
It's perfect. Yep.
Okay, well, great. Thank you so much. And thank you to everyone that's here, and also to the introductions given by Ashley and also by Gabby. So my name is Dale Lappinghouse, and I'm an associate professor at the University of Florida, located in Fort Lauderdale at the research center we have there. And I will get a little bit more into the weeds now on some of the aspects that Angela spoke about, as in compounds or what do these algae what do they produce? Gabby talked a little bit about genetics, and Ashley talked about what are some of the aims of this work. So I do love algae. I mostly like the smaller ones, but you see in these 2 photos, the small ones can get very big. And so yes, so that's just showing there. So moving forward, I like those that have seen my presentations have probably seen this. Okay. And why do I think this is important? If you just think about if you've not seen phylogenetic trees, what I want you to think about is your genealogical tree. So at the bottom there, at this, I don't think you can probably see my let me see if I have where do I do a little spotlight? Nope. Anyways, deny. Where it says original supergroup in the middle, that would be you, for example. And then if you go up, would be your parents, your grandparents, et cetera, et cetera. So if you go all the way to what I'm trying to focus, if you look at these groups, like it says SAR, it says Heptista, starting on the left, Cryptista, Archaeplastida, Amorphia. What you're seeing is that algae are found in Archaeplastida and Cryptista and Heptista and TSAR, and also in the exudates. And you can see in the bottom, I have more of this detailed out, but they have evolved separately. They've evolved for millions of years separately. And what does that mean? That means they have different physiologies, different ecologies, different uptake capabilities, different needs. Ashley spoke about nitrogen remediation that they did, but it's because they need nitrogen to grow. So this is the importance of looking at species, because different species have different needs, and also different species produce different things. For example, if you look at Archaeplastida, which is the green in the middle, you'll see Glaucophyta, Chloroplastida, and Rhodophyta. Chloroplastida are the green algae, Rhodophyta are the red algae, and if you go all the way down to the first on the left, Serenophylla are brown. So let me go back to the green algae. So all land plants are a green alga that's modified. And we know that a dandelion, a centipede, a palm tree, and corn all have different needs by physiological needs. They can be killed differently with different pesticides, herbicides. They have different services. They provide different products. Soybean does not have the same products as centipede does. And so even though all these are algae, there's so much to still learn, and they have so many valuable products to give us. So how do we go about finding that? So I gave you some of the conversation of why algae are important. So they are primary producers of organic matter, so the base of the food chain, and they remediate waters, they provide oxygen. But the two highlighted ones here you can see is talking about contribution to the economic well, being by providing food and medicine and other products, and we'll talk about that a little bit. And then they're also a source of high value compounds, for example, lipids, pigments, carbohydrates, nutraceuticals. And in this little scheme on the right, it's actually it's a microalga, but it is a native Florida diatom from here. And so it's looking at the natives and the microalgae. We've already worked on some of the lipids that are produced by these, and I'll go into that in a little bit. But someone in the conversation asked, where can I find algae? What can I get as a supplement? Where can I find algae to eat? Well, you're doing it every day. So here are some of the economical uses of algae, the brown algae, which have alginates. So I won't go through everything, but if you are eating frozen foods and pastry fillings and syrups and bakery icings and relishes and cooked instant puddings, meringues, chiffons, oh goodness, chiffon, right? If anybody's French, salad dressing, pimiento strips, there's so much stuff found in food. And in dairy, you can also find alginates and whipped toppings, milkshakes, cheeses, and flans and custards, instant breakfasts. Is that thickening agent. In industrial settings, algae are also used for papers, coatings, adhesives, textile, boiler compounds, welding rods, cleaners, castings, in ceramics. And then also on the medical pharmaceutical aspect, they use them as bulking agents and capsules and tablets. A lot of the vegan food uses this. But that's the alginates from brown algae. There's also carrageenan from red algae. Okay, also some are the same. You find them in frozen foods and desert gels and jams and jellies and relishes and salad dressings. Again, whipped toppings, milkshake, chocolate milk, cheeses. Going back more, air freshener gels, tertiary oil treatment, cleaners, enzyme. Again, in the medical part, a lot of the laxatives that it's used, again, bulking agents in shampoos and toothpaste, you can find carrageenan. And then at the end, I'll just give another example from red algae, again, and that's agar. Which we use agar a lot in the lab as well. But anyways, again, in foods and meringues and candies and cheeses and yogurt, and again, in industrial, helping make bridges, electrophoretic media, like I said, in the labs, we use it in microtomy media, adhesives. So people are using agar every day, and these are from red algae. So besides thinking about food, about, wonderful porphyra, which is the nori, it's actually Paropia now, unless they change the genus again. That's used in these humongous, for nori kelps, that you can have kelp chips and all this. There are products that are high value that can be extracted from seaweeds. And now the question goes, what do our native seaweeds have that we can use? And that's the unknown still. I mean, we are starting to learn, but that's still unknown. So for example, I'll give you an example of some previous work looking with turf algae. So in this case of how we went through, and as Ashley did say that we're at the beginning of this, but just to have an idea of the processes and how we think on moving forward to discover compounds from these algae. So in this case, we were looking at turf algae. So this is a mixture. So a lot of times it probably will be a mixture of algae because a lot of them grow all together. So it depends, and that can be good and that can be bad because that's looking at polyculture compared to monoculture. But in this case, we had, and I have a couple slides on this, is, we looked at algae, grab the bulk seaweed, and that's that water. So the biggest problem with seaweeds or any algal-based system is that it is full of water, and dewatering is a cost. And I know that Andrew is much better at cost analysis, and he can talk about that later, or he can include those in his analyses. But let's say, for example, of what we've seen, 85% was water, 15% was dry mass. And among that, what we have found is between ash is about 47%, and that changes. Ash is high in marine systems as well because of silica and salts, or if you have diatoms that are fouling can cause ash. But other systems, like land-based systems, will have very little ash. So this changes. They might have more protein. And so ashes and protein, you can see there, those could be used for fertilizers, okay? Then you have the component of carbohydrates, of the carbs, okay, and celluloses. Those are good, like macro products, let's say. These aren't so much high value, but these are just looking at it for use as butanol or ethanol. Oh, and I wrote ethanol Portuguese and not English. So the ethanol, the different process between butanol and ethanol can be that's the different fermentation process. I'm not sure how many people, how many here have done fermentation with lactic acid and everything, but you use different Clostridium bacteria to produce either ethanol or butanol. And the benefit of butanol is that it doesn't corrode like ethanol. But anyways, so what you're seeing is that you have a humongous part of this biomass that can be used for ethanol or butanol production. And then traditionally in the past, the lipids were looked at as biodiesel. I'll talk a little bit of like 2 slides about that. But nowadays, when we're looking for high value, we're also looking at a lot of the PUFAs, at the unsaturated fatty acids, the omega oils, those that are very good and heart healthy, because we're trying to move away from using fish-based supplements where you can use algal-based supplements. And if you go to Whole Foods, you'll find vegan omega oils, and guess where that's from? That's from algae. So going into the okay, I gave you all these numbers. What happens? How do we get to there? So this is using some of these numbers we had before. But when you're grabbing 100% of the 80%, right? So you're grabbing all the biomass that you had, and let's go down. Let's just see. So how do you get there? How do you do this? So first you've got to, and again, these are all costs that are associated. You have to disrupt the biomass by mechanical or ultrasound, and then you have to extract the lipids because you have a lipid part and a carb part, right? Usually the lipids are very low. And how can we change the lipid part? We can stress them. And Gabby spoke a little bit about, and so did Ashley, about learning about different cultivation practices that may or may not lead to higher production of certain compounds, right? So in that case, for example, we're looking at in these turf algae, we found that out of everything, we had about 4.5% residual oils that could be made as biodiesel. We had a 0.5% as PUFAs, and then mostly we're carbs because there's a lot more carbs in these systems, and that could be used for these, like I told you, these ethanols, but also for other endpoints. Okay, and again, but within all that, you always have solids, and those solids can be used. They're full of nutrients. Ashley said they uptake nutrients, and that's the NPK. And guess what? Those are those fertilizers, and there are fertilizers on the market that are algae based. They're proteins. There can be diatom shells or other silica based, and a lot of other things. So you're already seeing that there's a lot that can be, besides the carrageenan, besides in your lipstick, besides in your bulking agents. You're already seeing other things that can be used. And this was some numbers that we looked at comparing turf algae to corn. And what I just want you to see is when we're looking at the difference between carbohydrates and fermentation and efficacy, you could go to almost 2,000 gallons per acre per year for algae compared to 160 for corn, about 1,800 butanol for algae compared to 160 for corn. And then besides that, you had a lot of biodiesel, a lot of lipids that were found in these algae. So again, if you look at the way top, biomass yield, so that's how much we saw that we could do 50 dry tons per acre per year of algae compared to 3.1 for corn. And these numbers are old, old. These numbers are older, and again, so these types of information are what can be used for the economic analysis that will be done with all what we find. So I talked about lipids and sorry, I got to stop a phone call from a family member. So we talked about the carbs and the biodiesel lipids before, but more high value lipids are these omega 3s, 6s, and 7s. And I mentioned because especially using as a health food, omega oils, and especially omega 7 is antioxidant. It can help in brain function. And so these are found in algae. Okay, and these are some of the examples of the omega oils. And in this case, looking at Florida microalgae, okay, this is just an example. When you're looking at the profile of this fatty acid, table 1, you can see all these different PUFAs, all these different fatty acids. And so if you look at the right on the mass percentage, we did notice that palmitoleic acid was about almost 53% of the mass. So again, and that is 53% of stuff you can put in your, well, I don't know if I would do it directly, but anyways, things that are marketable for those that want to invest money and market these. So very, very beneficial. And these again in this case was a microalga, but it was going to Lake Okeechobee, which you hear about bad algae, but finding a diatom, isolating it, growing it, changing culture conditions, and guess what? We have a very good native microalga found on Lake Okeechobee. I'm not telling you guys to go there and drink it though. Don't take this away from this. But besides lipids, we also have other types of high value compounds, which are pigments. And these are beautiful examples of just different colors. And the pigments, there's so many beautiful pigments. If you ever freeze and thaw algae, you can see. And in this case is actually data from Calurpa from Charlotte Harbor. So this is some work we're doing with Zach Smith on the pigments from Florida native algae. And what can you see here? I know there's a lot of names. So what I'm going to point out, you see there's a few, because if you go left to right, you see chlorophyll. The chlorophyll is the fucoxanthins. There is at the way end, there's lycopene, which you know from tomatoes. So in there you can see fucoxanthins, and those are anti-inflammatory. They're anti-obesity, they're anti-cancer. The chlorophylls you guys can buy it also in the supermarket. You know, they're good for skin, they neutralize toxins. The carotenoids, the beta carotenes, again, immune health, heart disease. So a lot of these exist, but most of the knowledge we have on these is due to microalgal work. But in this case, as you can see here already, some preliminary data, and Zach does not know I'm showing this because he sent this to me last week, because he's developing a new method of extracting pigments. So I think it's wonderful because we can do this with our different algae to see what pigment profiles they have, and then do an analysis of understanding when we, like the same analysis I did before of understanding wet weight to dry weight to percentage, and then we can get a number of an economic number with this. But also we can also understand with these pigments how they grow, what lights they use. So this can also help in cultivation because of different lights that can be used, depths, and everything that pigments are used for naturally and why they exist. So besides that, a new area and we do a lot of this with the blue-green algae, is mining the genome to new metabolites. And if those that I learned a little button that you do with PowerPoint about design. So forgive me if things are popping because I just learned that and I feel like an old person. Anyways, so we're going to start with sequencing. So Gabby spoke a little bit about sequencing on the barcoding. And so interestingly, I also have reds, browns, and greens, Gabby. You talked about all three. And so in these steps, we go from a squish where you go out in the field and collect, you have the squishy tissue and you just grind it all up and you grab the DNA out of it, not grab, but through extraction techniques, library preparation, sequencing, there's an Illumina and PromethION. So you're doing long and short read sequencing to close these genomes. And then you have several steps of analysis. And so it's like this. It's just ACGTs, all these analyses, all this computation of months of computation to pull these together, especially these seaweeds have large genomes. Then you can get to a beautiful table like this, where you have biosynthetic gene clusters. And this here is also a gene of a blue-green alga actually that's found in Florida. And if you're looking at these, like you see clusters. And what does that mean? That means that these algae, or this specific alga, has gene clusters known to produce certain compounds. And these compounds here are mostly for, well, there's some toxic compounds, but a lot of them are chemotherapeutic, anti-tumor, anti-obesity, etc. Even some of the toxin compounds can be used for standards for science. So in this case, this would be considered a super producer. Okay, and that's how you do it. But how do you get from okay, so we know that they may produce it, but how do we know they do produce it? And this is where we have other collaborations with some of my Greek colleagues, and they focus on the mass spectrometry. And so this is well, I don't know if they know they're working on this, but they do collaborate, and they will be helping us if they don't know. Anyway, so in here, and we're working on, again, some of the two first photos of algae that I showed. That's what we're currently working with them, because we don't have all the seaweeds yet, but now we can focus on that after we receive the seaweeds. So we're focusing with them on the bottom part. It's called the metabolomics. Okay, and how is that done? So this is done with very, very fancy chemistry, high resolution mass spectrometry with these very different types of of machines. You start by the same thing I told you, squishing and extracting the material, and then you put it through these different machines. And what we're focusing on is untargeted. Untargeted means that we do not know what we're finding. It's kind of like mass sequencing, and you get all these metabolites. And with those metabolites, you can make these molecular networks. So you can start putting all these metabolites together, and then trying to figure out that this metabolite is related to this metabolite, related to that one. We know what that one is, so these are probably that. So that's trying to do a discovery of novel compounds. And that discovery goes through, again, like I talked to you guys about a bioinformatic for genes, the same thing with chemistry. And what is nice is that they in the past couple of years, there have been several new, the MZ-Mine, GNPS2, Sirius, these are new databases that also Triantophilos that I showed before, that he contributes to these databases, where we can put the gene from the genome, we can have the compound, and then you can also elucidate a structure, a secondary structure. And so with the structure, then you can start doing in silico analyses of understanding that this compound will affect this part of a cell. And in this case, again, work with David, Petra, and Triantophilos, we have found, for example, in some blue-green algae here, microcolins, dolastatins, and rivularia peptides. And microcolins are anti-tumor compounds and immunosuppressants. Dolastatins are used in target cancer therapy. So, and dolastatin was originally found in the sea hare. So in a little slug thing. The rivularia peptides, those are serine protease inhibitors, and those can manage blood clots. So you see that they're, again, so we started with the basic carbs, lipids, fertilizers, NPs and Ks, but there's a lot more to be found in these algae. And like I said, as they are extremely diverse, have millions of years of difference in age. Well, 2 billion is about is our eukaryote, 1.8, I think, eukaryotes. And some of these around 800 million years, about 1 million years of difference in age. So you're talking about 1 million years to be able to produce different compounds in these different groups. So anyways, I just want to thank you for your time. I do have to be somewhere else, but my email is there. I can maybe stay on for a second, but Angela, you will let me know what is if I need to answer any questions, right? So I'm just glad that you can see this, and maybe you have an insight of what you can see besides the normal I'm saying things. You see there's so much more to be found, and it's extremely exciting.
That was awesome. Thank you, Dale. And we know
Yeah.
you need to run. So what we'll do is if you have a question for Dale, please type it into the chat, or you can tell me in a minute and we can connect you to Dale via an email so he can answer the
Okay.
question specifically, because
Yeah.
I know I can't answer probably any questions about your talk.
Well, if I can't answer, I'll grab my USDA colleague and the Greeks that that they didn't know that they were part of this project, Angela, Ashley, but now they are. It's official.
No, that was
Okay.
fantastic. Thank you so
Okay.
much, Dale.
Thank you so
Okay.
much. And then Andrew, I hope I gave you some data for you to put in your economic analysis. Great. Okay. Thank you, everyone, and thank you, everyone, for being here virtually. Have a nice afternoon.
Thanks, Dale. Okay, so now we're going to talk about the money. So Andrew, show us the money. Go ahead and let you take over here.
Okay, is everybody seeing a slide? Good?
Yep, perfect.
Okay, so I'm Andrew Ropicki. I'm an assistant professor in food and resource economics and Florida Sea Grants Marine Economics Specialist. So I focus on, all things economics related to, Florida's marine environment. So a lot of stuff with fisheries and obviously aquaculture. And the economic analysis we're going to do here really has kind of two pieces to it. First off is kind of a market analysis to see, what types of products. Dale talked a lot about some really interesting ideas out there for different things that can be derived. Kind of what might be a good fit for seaweed culture here in Florida. And then the second piece is what we would call like a pro forma enterprise budget. And basically the idea is there that similar to what they do in agriculture, where they use enterprise budgets to figure out, what the return might be to a farmer in any given year for growing corn somewhere in the Midwest or soybeans, we would like to do the same for macroalgae, but we have to do it a little different. We put the pro forma on top because we're talking about a brand new industry, and we'll have to really rely heavily on what's available in the literature and what we learned from some of the trials being done by others on this project. So that's kind of the two economic pieces. Okay. So really the major objectives of the economic analysis, examine the economic potential of different species. So we want to, as Gabby talked about, the idea of kind of identifying those 5 or so species that are most prevalent in Florida waters, looking at the different product markets, and then production strategy. We're really going to focus on 2 different ways of producing this. And on the economic side, what we want to do, this second point here is focus on opportunities where Florida growers could have a competitive advantage that kind of gives them a captive market. So U.S. aquaculture has a lot of wins. I don't want to say it like it doesn't, but if you look at global aquaculture production, we are a very small player here in the U.S. We don't produce a lot, and that's not a U.S. only thing. It's a lot of developed countries struggle to compete on the big industrial scale, commoditized aquaculture production that happens around the world. And there are several reasons for this. We've got higher labor costs, higher land costs, stricter regulatory environments. All of these things kind of create a competitive disadvantage in certain spaces. So we want to focus in on those areas where Florida producers specifically might have a captive market. And to give you an example, one thing we've talked a lot about shellfish aquaculture here in Florida and in the U.S. That's one of the big wins for U.S. aquaculture. And if you specifically think about oysters and clams, they are able to, they basically have a captive market here in the U.S., especially when we talk about oysters, and that the half shell market it's hard for other countries to compete in that space. You know, they can't get fresh product and high quality to the US half shell market. So it's kind of us in Canada. And that's one of the markets, one of the aquaculture industries where we've seen lots of growth and we've seen successes. So we want to look for those areas where not only can we do it here in Florida, but can we do it profitably? And then lastly, you know, estimate the economic feasibility of commercial production, employing what we know from past research and findings from this project. So that's really talking about the enterprise budget. Oops, sorry, would not have done that with my mouse. Okay, here we go. So the market analysis, we're gonna, as we said, examine the potential of multiple species, focusing on high value or segmented markets. I've given a few examples of things we might look at there, but that's not exhaustive. You know, there's other outlets out there as well. Some of these don't go into super great detail on exactly where the market might be. Like for instance, with aquaculture feed, Dale was talking about some of those pigments, and some seaweeds are showing promise in terms of having the pigment that is used to help when farm-raised salmon get the color that the market really wants. And that might be a place where Florida seaweed could potentially compete if you have that right pigment, because the artificial stuff that's used now is quite expensive. So just kind of an example there, but we're going to look at a lot of different opportunities. We're doing an extensive literature review, trying to come up with what's out there as potential avenues for Florida production. And then also we want to look at the production requirements. So some of these markets might have you know, very stringent requirements with respect to the phytochemical content of the seaweed, product form and refinement. There might be certain opportunities that are better suited for, you know, land-based controlled environment, either through a RAS type system or a flow through type system, versus kind of the open water lease grow out method. So and then yeah, the last point kind of gets to that as well, culture methods and that kind of required control of the growing environment. We are going to look at growing this on land, which is much more capital intensive, but that extra control over the growing environment might open up new marketing opportunities for the product. Okay, so the pro forma enterprise budgets, I already talked about this, but we're going to use what we learned from the other parts of this study to create these production budgets. And we're going to do one that's kind of a land-based production using wastewater from a RAS system or a flow through that's growing fish. So the idea here would be kind of helping some of our fish producers, aquaculture fish producers in the state that are using RAS or flow through systems. You know, fish create waste and you need to deal with that waste. So maybe there's a way to kill two birds with one stone. The idea would be here to kind of almost bolt a seaweed or macroalgae production system on the back end that could help clean the water before it's discharged, while at the same time creating new a secondary product for that producer. So that's one thing we're going to look at. And then also obviously the open water macroalgae production system, kind of similar to our clam and oyster leases. The idea of doing growing them on lease sites out in Florida's bays. And then we're going to look at several sizes of commercial operations to see if we, you know, notice any potential economies of scale if you go big enough with this. And then, okay, so, you know, these enterprise budgets are pro forma. I mean, we are focusing on an industry that doesn't really exist right now. And because of that, you know, we'll get numbers for people, but we don't want just one number, because it's based on a lot of forward-looking research and assumptions about what an industry might look like, what production might look like. So because of that, we're going to be add a lot of sensitivity analyses, looking at how changes in key production and marketing variables, things like output price, growth and mortality rates, input and processing costs, how changes to those impact the potential profitability of macroalgae aquaculture here in Florida. Because it's, you know, anyone, you know, Ashley did a good job of talking about the idea that it might show that this could potentially work, or it might show that it won't work, or at least won't work right now. But it's also important is if the numbers show that it does work, to let people know these are pro forma. They're looking forward. They're based on a lot of assumptions, and there would be a lot of risk involved. And then lastly, you know, there's the focus on the nitrogen assimilation. You know, currently there is at least not an easy mechanism growers could access to get paid for those environmental benefits. But we would like to examine the potential effects of payments if they were to become available. The nutrient assimilation, the idea of, you know, same as we have, you know, carbon credits, nitrogen credits, that kind of idea, some type of trading system. How would that impact the economic viability of production? So that's another part of this enterprise budgeting that we'll look at, is if, you know, we can, if growers can capture the value associated with those environmental benefits they're providing, what does it do to the economic viability of production? And I think that's it. So I'll go ahead and stop sharing. And any questions or anything?
That was awesome. Thank you so much, Andrew. Yeah, and then I think our next step is to sort of move into a general Q&A. I know there have been a couple in the chat already, but before we do that, I'm going to ask everyone who's on the call to just take a moment and respond to this little poll that we're going to go ahead and shoot up on your screen. It'll only take about 20 seconds of your time, but I'm going to launch it now. And if you don't mind to just click through and answer it, that would be great. We'll just pause for just a minute. Okay, we had an 80% response rate. I'll take that. I'll go ahead and close the poll now and sort of open the floor for any questions that you guys might have. You can just unmute yourself and yell it into the ether. Oh, Carrie, go ahead.
Hi, how's it going? This is Carrie Flaherty-Wally. I'm with the Tampa Bay Estuary Program, so I'm really interested in what you all are doing, and we're really interested in macroalgae patterns within the bay in general. So my question / comment, I think, is for Gabby. So you mentioned in your presentation that you're talking about doing a macroalgae guide. Is that correct? So I want to connect you with Sheila Scalero, who's our seagrass macroalgae expert on staff, and she's actually already developed a macroalgae guide for Tampa Bay. So you should connect with her, and maybe you can add to or collaborate on that. And she's also really interested in starting a macroalgae monitoring program using citizen science. So I think y'all will have a lot to talk about. She couldn't make the call, but I've already kind of pinged her and said that she needs to talk to you. So
You're amazing. Thank you so much. That is actually directly in line with what I need, and I would love to talk to her. Thank you so much.
Great.
It's perfect.
Go ahead, Oleg.
Thank you. Thank you, everyone, for the great presentation today. Very informative. Appreciate everybody's time. I was just curious, because as some of you know, I've been looking into this for a little while. Would you at all be interested in receiving some seaweed samples from Tampa Bay, from the old Tampa Bay, that you could run for analysis through, the nutrients, potassium, phosphorus, nitrogen, possibly the pollutants as well. I know I'm going beyond what was said in this presentation, but even if we could just get the base nutrients, that would be terrific. And the reason I'm asking is I paid for some of those samples to be analyzed myself a while back. It's quite expensive when you go to a private lab. And when I had approached UF in the past, they said, even if you were to bring us grant funding, we don't have the ability to go beyond our scope, our existing scope for looking at soil samples and other plant samples within the state. Like basically they have to stay within their lines. And I'm just curious if through this study, you might have the ability to expand a little bit and review what others might be able to send you. And last thing I'll say, I'm right off of Old Tampa Bay, right off of Bay Shore. And so I could send samples pretty readily. And I think comparing, seaweed that grows close to a high density population center versus seaweed that Aaron has in his farms that are at the mouth of the bay, that might be interesting in and of itself, the capacity to absorb the nutrients. Thank you.
Ashley, I think you should probably field that one.
I was just trying to find the unmute button. Yeah, so we can talk offline and figure out how we might be able to get some of your samples into some of the queue for what we're doing. But we don't have a lot of capacity to add on very many samples, but I think that the idea of comparing is definitely something we want to explore. So it's very relevant to our questions. So I'll type my email into the chat and you can shoot me an email. We can talk a little bit more.
Thank you very much. Appreciate it.
Yeah, and on that note, I think, Oleg, we should talk offline as well, but I think it's important to note again that this is just like the first of hopefully many projects that continue to explore some of these different avenues of research. And these collaborations that we're identifying early on are so helpful to inform like the future work, right? So even if there's not money right now for those samples, perhaps it could be like a grant proposal or some sort of a collaboration where we can find a way to finance that. So let's just keep that conversation going.
That would be terrific. And just for everybody's, so everybody's on the same page here. I had an interested funder in one of the big US foundations, but the folks, and I need to go back to the names of the folks I was exchanging emails with, and it was actually Tampa Bay Estuary Program said you should reach out to them and ask if they could analyze your samples. They said, look, even if you bring us the funding, we can't carry out the analysis because we have our work program for the year and we're not allowed to deviate. That was then, but that was more than a year ago. Obviously if there's a little bit of space here, I'd be happy to reconnect with the funder and see if there's still some interest there. And so I wouldn't need any of US funding to do it. I would bring the funding for the samples to be done.
Yeah, we can definitely chat later. I'm pretty sure my lab is just going to turn into like a seaweed storage room and processing facility pretty soon. So
Yeah, thanks for that, Carrie. For those that aren't familiar, the estuary programs in Tampa Bay and also Charlotte Harbor do eyes on seagrass surveys that also include macroalgae assessments. So there are data that are available regarding macroalgae cover in certain estuaries in Florida, which I think can also help kind of fill out and round out the information that we have on what's already occurring. Those surveys aren't specifically over shellfish leases, which is one of the differences with this project, is we're focusing primarily on the algae species that are aggregating over shellfish leases and active shellfish growing areas, because we know that an industry already exists in that space. And there is a mechanism currently for some sort of harvest from that space for human consumption. That's going to look a little bit different for seaweed than it would for a bivalve. But at the same time, like there's already this commercial operation in place on the leases, which is why we chose that rather than looking at like every macroalgae that occurs in Florida, because that like fitting into the aquaculture industry might be a bit more of a stretch. So this was just like the first step in the process of identifying which species are the most abundant at aquaculture operations that are already in play for a different organism.
Yeah. To echo that, we thought like these are permitted. So if we get any material collected, these locations have already gone through the official permitting process. And then there's already permitted for gear too. So it's a little bit of our attempt to figure out what can attach to the gear that we already have that's been able to get permitted for leases, and without having to modify and like come up with some new way to grow the macroalgae. I do think that comparison though of what does accumulate within on those leases versus like what might be found nearby in Tampa Bay is really interesting to see if we're getting any of those. There's different species.
Yeah, I'm pausing because I'm looking back in the chat because there was a question about macroalgae and ocean acidification comparison. So I didn't know, Ashley, if you wanted to speak to that or not.
Sure. Yeah, I think that there's a lot of opportunities like for seaweed culture going forward, and their functional role in these ecosystems does go beyond just that of water quality restoration, has some climate mitigation potential. Not to go back to this, but budgets are kind of limited. So we opted to focus on the water quality restoration angle. And part of that is also because on the East Coast of the United States, our real water quality issues and pressing our issues are related more so to nutrient loading, less so to ocean acidification like we see on the West Coast, because just of the way that our coastal system works, and like we're much more connected to land and don't have that like big upwelling. So ocean acidification is much more of an issue on the West Coast, and eutrophication is much more of a pressing issue on the East Coast. But opportunity to expand for sure.
Are there any other questions out there? I saw another one of the early questions in the chat was, where can I try seaweed products to eat? And there are actually a ton on the market, just none of them are farmed in Florida. So obviously, because we don't really have a market yet, but there are lots of ways you can try seaweed in lots of different varieties.
I was gonna say really quick, the best part is that some of them are actually, I don't know how close you are to a Sprouts or Whole Foods. Obviously, those are the pricey ones, but they have been experimenting a lot with having seaweed-based products in their stores. So definitely go look around and see anything, if you can find anything, versus, you know, Publix or something like that has more of like those like little crunchy, like, I don't know how to describe them, but you know what I mean.
Snacks.
Yeah, the seaweed snacks. But yeah, people are starting to get creative and looking into those markets, and you can always order something offline too. Like I'm a big proponent of kelp pickles, but that's just me.
Okay, there's a question from Emily, and I think that Andrew might actually know the answer to this question right off the top of his head, but she asked if there's an IMTA, if there's any research on IMTAs with finfish or wrasse in Florida currently.
Yeah, I believe we have a new red drum farmer in, I want to say, Lee County, who I'll have to get back to you with specifics, but I think they might be. I'm not sure if it's seaweed or if they're growing some salt tolerant plant, but they do have a multi-trophic aquaculture setup.
Yeah, I think that's the sorbies with Pine Island redfish, and
yeah,
I think
Island
it is, yeah, like a sea vegetable.
Yeah,
One of those. But
you're right. It's not a seaweed. It's a yeah, a little different, but yeah, kind of that idea.
Okay. Okay, well, you guys, I just want to remind everyone too that we will be sending you a link to the recording, as well as try to compile some of the relevant resources that we've just talked about, and give you those links so you've got them at your fingertips. You can also reach out to any of the individuals you heard speak today individually if you have a specific question for that person. We've got a couple of different questions here in the chat about people that want to start seaweed farms and are interested in entering the industry, and that's fantastic. But I just want to remind everyone too that we're not there yet necessarily, right? So part of this project is to get some of the data to inform future industry incentives in Florida. There are definitely mechanisms to enter the industry if you're interested in it, but that isn't something we can provide guidance on necessarily at this moment in time. There is a seaweed hub that is, national, and it provides a ton of information for different states that are culturing seaweed currently. And I'll provide a link to that when I send out the resources, and I'll also stick it here in the chat in a moment. But I just don't want to think we were ignoring your questions about wanting to start a seaweed farm. We see you, we hear you, but I don't know if we're the best group to provide information on how to start a seaweed farm quite yet. But Andrew, Ashley, Gabby, tell me if I'm incorrect in that.
That was great, Angela. Thank you.
Oh, thank you, Emily. Emily stuck the Seaweed Hub link right there in the chat. Okay, well, pending no further final thoughts, I'd like to really thank everybody for joining us today and hearing about this project. It's just beginning. You'll be getting information from us as we move forward, as results start to come in, and there will be, I'm sure, another one of these to present project results in a couple of years. In the meantime, please don't hesitate to reach out to Florida Sea Grant or the University of Florida for any questions that you might have about any of the stuff that's going on. Thanks for coming.
Thank you so much for all the time today and the presentation, guys. Appreciate it.
Thank you. And thanks to all of our speakers.
Yes.